antibodies are also called as immunoglobulin. Immunoglobulin, globin means, you understand by the very word, it is a protein. Okay, globin means it is a protein, and immuno means which is produced from our immune system. So, antibodies are protein substances which are synthesized by our immune system, or they are produced by our immune system. And when are they produced? They are produced when there is an attack from an infectious agent or from the foreign agents. A foreign agents can be any type of microorganisms, or I told you even an allergen or a chemical or a toxin. These all can be your foreign substances, and in one term these foreign substances are called as antigen. So, usually what happens is, now suppose say like we will come to our same picture, if this is our microorganism, okay. So, what we told, our cells, our immune system is not going to recognize the entire microorganism, instead it will be recognizing only the antigens, and that antigens also it is going to recognize only the part or a portion of that antigens which were called as the epitope. Okay. Now, what happens, remember our antibody, okay, our antibody is always how it is in Y shape. Okay. So, I will just draw this antibody. Okay, this is my antibody and it is Y shaped, and it has to fit exactly into this epitope. Okay. Once it is bound, then what happens, this antibody when it is developed in your body, in your bone marrow, it is always a naive, that is it is in an inactive form. Only when it binds to an antigen it will become active, otherwise it is naive. Okay. This antibody when it is synthesized in your bone marrow, it is actually naive, that is it is inactive form, but when it keeps on circulating in your body, and it is acting like an army safeguarding your body against any microorganism. Once it encounters a microorganism, then it becomes activated. Okay. Then what it does is, see now what happens, your antibody has gone and coated on an antigen. Okay. Where is the antigen? The antigen is the microorganism. So, now this has become coated with the antibody. Now, what is there? This is now the antibody. Okay. Now, what it will do is, it will present this, okay, this antibody which has coated your antigen, it will present itself to a complement. Okay. It will go and it will present this to another type of proteins called as a complement. We will just come in a second, we will come to know what is the complement. For time being, just remember this. So, my antibody has gone and bound to the antigen, and then what it will do, it will just present it to another type of protein called as a complement. Now, these antibodies, in today's class we will see about the complement and how these antibodies are produced. So, these antibodies are produced by a specialized white blood cells called as B lymphocytes or B cells. So, what happens? We know that our body has the white blood cells. Now, these white blood cells, okay, these are the stem cells from which different cells can be developed, or they are produced, they undergo maturation into different type of cells, and one type of cell is your B cells or the B lymphocytes. Why it is called as the B cells means it was first identified, these antibodies were first identified in the birds, okay, in an organ called as bursa. Okay. B-U-R-S-A. Okay. In the bird it was found first, and it was an organ called bursa in the bird where the B cells or the antibodies were identified. So hence even in the humans they found that it is similar even in the humans, and they also called it as a B cells. So now what happens, as I told earlier, whenever a body encounters an antigen, this B cell, okay, it will come and it will notice that there is an enemy. Then what it will do is it will divide and it will mature into identical cells called clone. Okay, that initially in your body there will not be many B cells, but once it is triggered by the antigen, once it is triggered by your enemy, then what it will do is this particular B cells will divide and it will mature and develop many number of cells, okay, millions of cells it produces, and these cells are called as plasma cells. Since they all appear to be same in appearance, they are called as clone. Now what is the function of your plasma cell? The function of my plasma cell is to produce the antibodies. Okay, that's, remember the first slide how we defined our antibody is. So our antibodies can then go and bind to the cells and they can bring about the positive reaction. But remember, first always your B cells, okay, they were naive cells, they will not be knowing like because they have never been encountered any antigen in the beginning, hence they are called as the naive cells. But once they come in contact with the antigens, then they will become activated, and the process of activation you will be studying in the next slide, and it is an important question for the examination wherein you have to draw the diagrams also for this. So once the antibodies are produced, they will be released into the bloodstream and the lymphatic system. So if the antigens have, or the microorganism, once it has entered into your bloodstream or even into your lymphatic system means where the lymph nodes are present. Okay, you can sometimes feel whenever you are having fever and all near your part of your neck. Okay, that region sometimes there will be swelling, okay, severe fever or any infection, okay, because these are the region, particularly your neck and your stomach in the sense intestine, where there are lot of lymph nodes which are present. So these lymph nodes, what they will be having all your immune cells which are present. Okay, so what are your immune cell? One of my immune cell is the antibodies. So once the antibodies are produced, they will be drained into the bloodstream or the lymphatic cell where they can encounter the antigen and they can deactivate this antigen and throw it away from our body. Now we just told in the slide that the antigen coated antibody, what it will do is it will present itself to a complement. What is a complement? A complement is a complex system, okay, like these are like a a concert of proteins. There are at least 30 proteins which have been identified, and what are the function of this complement is, their function is to eliminate the infectious microorganism. Okay, so how they are bringing about the elimination of the microorganism? They have three functions through which, or we can say there are three mechanisms by which the complement can eliminate the microorganism. The first one is called as lysis, or it is called as the bursting. What it is going to burst? It is going to burst, or it is going to lyse my foreign or the infected cells. The second one is called as phagocytosis. We have studied in detail when we have spoken about innate immunity. Once again, this is an important question for the examination. You have to write the mechanism of phagocytosis. Okay, so the second is by the phagocytosis, that is it will ingest the microorganism or the foreign particles or even the cell debris, our own very cells. Okay, they have fought against the diseases and they themselves would have died. Okay, those cells debris are also eaten by the phagocytosis. And the third method, which I have already explained again in the innate immune system, is the inflammation of the surrounding tissue. When I told you, whenever there is an attack from the microorganism, you can see that there is an inflammation, okay, there is a pain, there is a red color, there is swelling, okay, indicating your body, it's telling your body that I am severely infected by some microorganism. All other cells become active so that we can fight against the particular infection. So that is a classical phenomena of inflammation. Is you will be having the fever, you will be having pain, the rednesses, the swelling, okay, all this is called as inflammation. Now we'll look into each of the step. So what is a complement? A complement is a complex of protein. Okay, this protein, from where actually this complement is produced, means it is produced in your liver cells. Okay, we have liver, right? From the liver these complements are produced. So what are these complements? It is nothing but a protein, and what they do, they keep circulating in your extracellular fluid in an inactivated form. Even these, they just, if this is, suppose this is a person, okay, what happens, this complement system is just circulating throughout his body, okay, whether there is infection or not an infection, it keeps roaming about. But here what, it is an inactivated form. But suppose say there is a pathogen, okay, then what happens, it becomes active now, and once it becomes active, it uses all these three mechanisms to evade our pathogen. It will, okay, it will deactivate or it will kill our pathogen. So we will see this all steps in the next slide. So this complement, this group of proteins, this group of 30 proteins, okay, their activation, we told that they are inactive. If they become active, there are two ways in which they can become active. One is called as a classical pathway and other is called as an alternative pathway. When you go for your higher studies, you will be studying in detail about the classical and alternative pathway, and this is also an important question for your PGCET, for your MSc or GATE or any other exams when you take. So a complement activation occurs by two routes. One is the classical pathway, the other is the alternative pathway. I'll just put the next slide and show you. See, in classical pathway what happens is, this is a microorganism, okay, and see, the microorganism has certain protruding region. These protruding regions are your antigen. So now what happens, once the body is encountering this antigen, immediately your antibodies which were circulating, okay, these antibodies become activated once they face the challenge from the microorganism. And now what happens, I told you that the antibodies are Y-shaped, and this is the region which they bind to the epitope, and the region of the antibody is called as the paratope. Okay, this is the paratope and this is the epitope. Epitope is the region of the antigen and paratope is the region on the antibody. We will be dealing in detail in the next class about this. So this is the Y shape and this is the stem of that Y shape, right. Now the stem, where it is gone and bound, it has gone and bound to something called C1 here. Remember, the C1, it is nothing but your complement, which is represented as C. Okay, it is represented as capital C, means it is a complement. What is a complement? It is a protein which is circulating in my body. How it is, where it is produced? It is produced in the liver. It is present in an inactivated form. But when there is an attack from the microorganism, my complement can become active. So how it can become active? It can be activated by two pathways. One of the pathway what we are learning is the classical pathway. In classical pathway, Look at the definition. The classical pathway is considered to be the part of the specific immune response. Be careful, specific immune response, because it relies on the antibody to initiate it. What is specific? When we told specific are adaptive immune system. Okay. So, in adaptive immune system means what has to come to your mind? T cells and B cells have to come into your mind. So, now what is happening? This complement system is becoming activated. When it is becoming activated? Only when the antibody is bound to the antigen. That is called as the classical pathway. Hope it is clear. Just go through the definition. Okay. The second method is called as the alternative pathway. The alternative pathway is non-specific defense. That is, it does not require any of the B cells to get itself activated. It does not need the antibodies or the B cells to itself to get activated. So, what happens is, if there is a microorganism, there is no this antibodies. Okay, directly your complement can go and bind to the epitope of the antigen, or it will go and bind to the antigens of the microorganism, and it can bring about the denaturization or killing of the microorganism without the help of the antibody. That is called as an alternative pathway. So, remember this. Alternative pathway A, it does not require any antibody, it is independent. So, since this alternative pathway, it does not require any antibody, it has to do its work by itself. So, it is slower in the process when compared to the classical pathway. So, usually whenever we write a complement protein, it is written in an uppercase, capital C, and they are always an inactive form unless they encounter the antigen. So, now what happens? You will see this picture. Once they encounter, whether it is classical or alternative, once they encounter the antigen, what it becomes is, it gets activated and then it breaks itself. Okay, this complement systems, then they break themselves into two parts. One part will be bigger, okay, and the other part will be smaller. For example, here I have written like the C1 is broken into C1A and C1B. Similarly, for example, it is a C3 mean. C3 will be broken down into C3A and C3B. So, this A is the smaller part and B is the larger part. Okay, that was C here. Okay. So, it will be broken into two different parts. For example, here they have shown C2, it gets broken into C2A and C2B. Now, what happens? Now, we told there are many proteins. Now, if activation of one complement will trigger the activation of all other proteins. Okay. So, now what happens is, like you will see that what to say, you just make a stand of book. Okay, let all the books stand by itself in vertical shelf. Just push one book, then what happens? All the books will fall, right? That is called as cascade of reaction. So, here what happens, if one protein gets activated, immediately the next, third, fourth, fifth, immediately all gets activated. This is called as a cascade of reaction, or it is also called as the chain reaction. Now, this entire picture will tell you what are the three mechanism or the function in which the complement is defending our body. So, you can see girls, here this is the classical pathway and this is the alternative pathway. So, here what happens, classical pathway, my antibody should bind to the antigen of the microorganism, then it forms a complex antigen antibody complex. In the alternative pathway, I do not need an antibody, directly my cell or my complement will recognize the antibody. So, now what has happened? Okay, both, whether it is a classical or alternative pathway, they have one meeting point. one converging point, okay, that is called as the C3. C stands for complement free protein. That is the numbering in which they have been identified. The scientists have given them the numbers. Okay. So now the C3, what happens is it has to break itself. Okay. So now this C3 will be broken and now it is represented as C3A and C3B. So what is the function of your C3A and C3B? We told it has three important functions. One is the cell lysis. The other is the phagocytosis. Okay. The third is the inflammation. So you see here the C3, first we will look into the opsonization. Okay. Here you see this, the C3, what it has done, it has encountered your pathogen and then it will go and coat the pathogen. All the C3 proteins, many C3 proteins will go and coat this pathogen. So what is happening? This process of coating of the protein onto your microorganism, it is called as opsonization. Okay. By complement, it is going and coating on the microorganism. This is an opson, which the process is called as opsonization. That is what is happening is it is acting like a tag or it is like a marking telling other cells, okay, now if this fellow was like this microorganism as such it was means maybe it would have escaped our immune system. But now once it is being marked, okay, so now in a class of 5 if you all are sitting and I don't, from behind if I see you all, I am not able to identify you all, but someone is having a hat on their head, okay, or some ribbon, then I can identify that particular person is different from the others. Similarly here what happens, it is being tagged, this microorganism. Then immediately what will a macrophage will do, it will come to that site and it will start engulfing our microorganism and it will kill the microorganism. So this is one method. Next what we told, will come to inflammation. Okay. In inflammation, what is happening? What causes the inflammation? Inflammation is actually caused by one of the cells called as the mast cell. For example, this is a mast cell. Okay. This also you all studied in innate immune system. So once this mast cell, inside the mast cell what are there? They are histamines are produced. I told you histamines are chemical agents which are present inside my mast cell. Okay, they are enclosed sitting inside the mast cell because they are very harsh chemicals. Now what happens once they find out there is a microorganism, there is a pathogen, immediately the mast cell what it will do is it will release all its chemical, and these chemicals what they do is they can go and attack the microorganism, or what they do is they attract who? They are going to attract our phagocytosis, okay, the cells which can eat these microorganisms. So once a phagocytosis becomes activated, then they are going to ingest this microorganism. So this is another method. The third method, you can see the next slide, that is the lytic pathway, this one. Okay, you see in lytic pathway what has happened, there are many more proteins which have come into picture, C5, C6, C7, C8, C9, all these proteins have come into picture now. So here what it does is, all these proteins, okay, from how these proteins are formed, these are all complement proteins, okay. The initial protein was C3 protein. C3 protein, it is getting broken each time and it is activating C4, C5, C6, like that, it keeps on. So now what these all will do is, all these proteins, this is your microorganism, this is the plasma membrane of your microorganism. Suppose say this is the microorganism, this is the plasma membrane, the outer layer is your plasma membrane. That is what in detail they have shown that. All this protein, they will form what are called as membrane attack complex, okay. So this is all your protein, they will form a complex around your plasma membrane and they will make a hole. On who they are making the hole? They are making hole in the plasma membrane of the microorganism. So there is a what? There is a gap. Entire thing only went up. Okay. Now they will bore a hole into your microorganisms. Now what happens? All these cells will go and attack it, and they release all their chemicals. Now what is happening? Since there is a hole, all your immune cells will enter into it. All your good cells which have fighting against the pathogen will enter into it, and now what happens? Your microorganism will start swelling. Okay. Once they swell, immediately they will burst. So this is called as cytolysis or the simply lysis. Okay, that is the breaking or the bursting of the cell. So loss of cellular content through transmembrane channel is formed by the membrane attack complex, that is from C5 to C9. They go and sit on the plasma, they will bore hole into the plasma, so that all my phagocytes, my dendritic cells, NK cells, all other fluids will enter into it, resulting in the swelling of my pathogen and ultimately bursting of my pathogen. Here is an important point I have put. Just remember these three keywords. One is the activation. We told that in my other slides also I told you there are different types of antibody, particularly there are 5 types of immunoglobulin. Okay. So IgG and IgM are the antibodies which can activate the complement system. We will be dealing this in the next chapter. Okay, in unit 2 in detail we will be talking about this. Okay. So just for time being, remember, of my 5 immunoglobulins, IgG, okay, that is gamma and mu, can activate my complement system. So what is amplification means? Many of the membrane attack complexes attack, punch holes in the walls, resulting in the cell lysis. So this is how my complement system, with or without the help of the antibodies, it is killing the pathogen and throwing away the pathogen from my body. So coming to the next slide, we will now just what we spoke in our first slide. We spoke about the antibody. We spoke about the antibodies are produced by the activated B cell. Now if you are speaking about the antibodies, now I should tell you like how these antibodies are produced, how they are getting activated, how you are telling that the clone of cells are being produced. That we shall look it into this slide. See, once these antibodies are produced in your body, right, and once they throw away your pathogens, it is not that they will immediately go under resting. Okay, they will remain for some months in your body. They keep circulating. Okay, now we'll answer these questions what we asked. This is an important question, and this is your topic of today's assignment. You should write about what is the clonal selection in detail with the diagram. So we have this naive B lymphocytes. Okay, these naive B lymphocytes, how they are produced in the body? They are produced by the stem cells. Where are these? These are present in the bone marrow. Why these are called naive cells? Because they have never encountered an antigen or never been challenged by the antigen. So when there is an antigen, how they become activated? How there is a clonal selection? How there is clonal expansion? That is what we are going to study now. So now what happens is, this naive B cells in the bone marrow, they will be genetically programmed, okay, to form into an antibody. So usually what happens is, each of the B cell, okay, now whatever B cells are being produced by our B lymphocells, they are unique by themselves. Many type of B cells are produced, but each have their own uniquenesses, they have their own specificity. Each of the B cell produce unique antibody. How they are produced? They are produced by what are called as genetic programming. What is this program? Okay, that is, there are genes which tell them that you have to get what proliferate yourself so that you produce more number of cells. You proliferate your cells into different type, carrying different type of receptors so that it can bind to the different type of antigens which our body can encounter. So this is called as programming. And who does the programming? It is done by the genes, and this process is called as gene translocation. Look at the definition. Clonal selection during its development. Whose development? The development of the antibodies from your B cells. During its development, each of the B lymphocytes, okay, these are all your B lymphocytes, they become genetically programmed through a process called as gene translocation to make a unique B cell receptors. Okay. So, they become genetically programmed by a process called as gene translocation, so that each of the B lymphocytes now become B cells carrying receptors. Okay. Each, you look at this pictures, each one is carrying different type of receptors. Okay. This is called as clonal selection. Once now a B cell carrying different receptor is produced. Now next step in the B cell clonal selection is one B cell. Okay, suppose say here in this diagram we have taken this B cell. Okay, a B lymphocyte which can appropriately fit into a particular antigen. Okay, that means I'll draw it in a simple diagram and show you all. There will be, like, for example, let me change the color. Suppose say I'll mark it in a, this is all my antigen, you'll think. Okay, these are all my antigens, and come back to the picture. This is all my different, okay, one second girls, that is this eraser, and okay, this is all my antigens. Okay, and this is my this is my B cells. These B cells are what? There are clone of cells carrying their unique type of receptors on them. Now this is a B cell, this is a B cell, they'll be all circulating around, okay, around this antigen, but every B cell cannot bind to this antigen because they are not having any specificity. A B lymphocyte with an appropriately fitting B cell receptors can only bind to the antigen. Now suppose say this is a blue antigen, there is nothing like blue antigen, just an example I am giving, a blue antigen. So this and this is also blue. Now what will happen? Okay, there is an exact fitting between this. Now they can go and bind to this. That is what we have shown here. Okay, so there is an virus, this virus is having an epitope. Now if there is an affinity between our B cell receptor, that is, it can exactly fit into the epitope of this antigen, then it will go and bind to it. This entire step is called as the clonal selection. See, now I don't need this B cell 1, I don't need this, I don't need B cell 3, I need only B cell 2, because that has recognized my antigen and that should have do its duty to deactivate my virus. We will come to the next slide. BCR means B cell receptors. How they are produced? Because they are programmed, this naive B cells are programmed by who? They are programmed by the gene translocation. Okay, it is a gene which tells them that you get converted or you get modified to a respective receptors. Okay, now they have bond. Now what happens? There should be a clonal expansion. One B cell receptor cannot go and attack the n number of antigen which has entered into your body. If there is an 100 viruses, there should be more number of my antibody, just not 100, more number it should be, so that it can encounter that, it can go and build its army against our enemy and fight against it. Now that step wherein it is going to expand, it is called as clonal expansion. Now here what happens, we told that our antigens are T cell dependent and T cell independent also are there. So this is what? T cell dependent. For example, these are what? T cells. What it is written as T4 lymphocytes. T4 lymphocytes means these are T helper cells. Don't worry about all this, because in detail you will be studying, I told you already. Now what is this T helper cells? These, what is the function of T helper cells? They activate the cytokines. Now these cytokines, they will send a message to this B cell 2. Now there is a pathogen which you can go and bind to it. Now what you do is you just start multiplying yourself. That phenomenon is called as proliferation and affinity maturation. From naive cell it has become matured now. Okay, it has become matured now, and you can see that from few cells I will get many type of B cells. Okay, the cytokines from an affected T4 lymphocytes now enable the activated B lymphocyte to proliferate into a large clone of identical B lymphocytes. This will come for 2 marks. What is a clone? During this time, fine tuning of B cell receptor occurs through the affinity maturation. Now they are finely formed that they can go and exactly bind to that particular type of antigen which is going to cause an infection in me. Okay. So now how many? Many cells, B cells are formed. Now these B cells, they should undergo differentiation. Okay. So see the next part. Differentiation of B lymphocytes into what? What are you going to get differentiated into? I will get differentiated into plasma cells and B cells. Okay. So now you can see this, these are all these B cells. Okay, I put all these B cells here, and these in turn, okay, this is also our B cell. These B cells will then get differentiated into plasma cells. Okay, and they also get differentiated, some of the B cells get differentiated into B memory, not one memory. This is just I've drawn one picture. Many memory cells will be produced, many number of plasma cells are also produced. So what is the function of the plasma cells? The function of my plasma cell is to produce the antibody. Can you all see this blue color? These are all your antibodies. Now what is the function of the antibodies? The function of my antibody is now to go and counter attack all many of the antigens, that is the viruses which were produced. Okay, see here it encountered only one type of antigen, it studied about that particular virus, then it proliferated itself, it matured itself, it differentiated itself to produce many antibodies so that it can protect us from that particular type of antigen. This is all about the clonal selection. Okay. So usually the same thing in I'll just explain in brief. Okay. Suppose say if there is a bacteria, okay, the bacteria has entered into your body, then what we have, we have the lymphatic cell system in our body, that is the lymph nodes. The lymph nodes are the ones which immediately come for your protection. Who's inside the lymph nodes? There are different immune cells which are present. Now what we are talking about only the B cell maturization. Girls, this is the same how it happens even in the T cells. When we are talking in the T cells, we will come to it. This is how the T cell proliferation also occurs. I have just given here the example of B cells. But what happens is T cells does not produce antibody. They produce T effector cells, and similarly they produce the T memory cells. So inside that lymph nodes, what now your blood and all, what it will do is it will drain this bacteria into the lymph cells or the lymph nodes. Now the bacteria has come into your it has come into your lymph nodes. Inside my lymph nodes, what is there? Now we will think of only about this is the bacteria. Now what is there? Many types of B cells are present. Okay, inside this many types of B cells are there. Okay, different type of B cells are there. But by chance what happens, this bacteria and this B cell, okay, they will find that okay, we can exactly fit into each other. So once they find out this, now what happens, this B cell, okay, now this B cell has to undergo clonal selection. This is designated to undergo clonal selection. Okay, that is what is all this about. Then it will develop the B cell receptors. Then it will go and it will get proliferated into number of cells. Okay, now this cell, this will become more in number, and now they can go and bind to the bacteria and then can kill this bacteria. And then what happens, whatever the left out parts of your bacteria or the dead antibodies are there, they will be chewed up or they will be ingested by your phagocytic cells and they will be thrown out of the body.